the official start of the AGA, and we have a terrific program, I think, to get things rolling. And the session is on the biology of preneoplasia and why do some tumors become bad. And we have 4 speakers today. I think the way we're going to try to do this is ask the speakers to keep to about 20 minutes. There'll be no questions after the individual talks, but then we'll ask all 4 speakers to come up, and then we'll have hopefully 10 or 12 minutes to have general questions. And so I'm moderating this session. I'm Bob Coffey, and I'm moderating it with my colleague, Robert Wong from Stanford. And we're going to ask each of the speakers to keep to the time of 20 minutes, and we'll get started with Robert giving the first talk.
Good morning, everyone. Welcome to DDW 2025 in beautiful San Diego. I'm Robert Huang. I'm an assistant professor at Stanford University. And the talk I'm going to be giving today is a topic near and dear to my heart, gastrointestinal metaplasia and progression. Here is my disclosures information. So I wanted to start with this slide showing Correa's cascade. This is a cascade that many of you know well. And so with normal mucosa, we begin with some source of inflammation, either exogenous due to Helicobacter pylori infection, which is believed responsible for some 70 to 80% of cases of gastric cancer worldwide. But in the United States and other lower incidence nations, there's also a high prevalence of autoimmune gastritis and autoimmune directed attack against the parietal cells of the gastric corpus. This leads to chronic gastritis, chronic atrophic gastritis, intestinal metaplasia, dysplasia, and finally gastric cancer, named after Pelayo Correa, which initially began as a hypothesis, is now fairly well accepted in the scientific community as the correct biological pathway toward gastric adenocarcinoma. So the topic of today's talk is going to be gastric intestinal metaplasia. So you might ask, why focus on GIM among all of these other potential precursors to talk about? So I think from a gastroenterologist standpoint, there are a couple of reasons why this is an attractive target for intervention. Namely, it is readily detectable by endoscopy. There's less intra-observer variability when assessing GIM histology compared with some of the other precancerous conditions seen on this slide. And finally, it's less likely to resolve following Helicobacter pylori eradication therapy. There have been some modeling studies as well as some observational data that have suggested this. So what is the overall risk of progression from intestinal metaplasia onto adenocarcinoma? In lower incidence Western nations, the best data we have is derived from European nations with robust population cohorts. So this is registry data from Sweden, published in BMJ in 2015. So they had a national pathology database, which was linked to a national cancer registry. And the red line, the dashed red line is intestinal metaplasia. You can see that the overall incidence of progression is actually not that high. Over 15 years, just about 2% progress. What do we know in terms of clinical risk factors for progression? Well, actually, we know a decent amount. We know that extensive IM, which involves both the antrum and the corpus, is more likely to progress. We know that histologically severe IM, that is, when there's a higher percentage of glandular involvement in the microscopic field, that's more likely to progress. And we know that incomplete IM, that is, IM with colonic phenotype, with variable mucin size, with a loss of the brush border, this is more likely to progress. And the extension and the histology can be captured nicely in an aggregate score called the OLGIM, or Operative Link for Gastric Intestinal Metaplasia Staging System. It seems to be this is the best kind of clinical tool that we have to risk stratify patients for progression. So you might ask, if we have these good clinical risk stratification tools, what is the role of biology? Do we even need to really understand the biology of this disease? Well, I think there are 2 reasons why enhanced biological understanding of metaplasia would be helpful. The first is, I think we need to understand these distinctions, these clinical distinctions on a glandular and cellular level. And secondly, we need to ask if we can enhance on the existing clinical risk stratification using molecular data and biological insights. The first study I wanted to present today is the Singaporean GSEP, or Gastric Cancer Epidemiology Program. In my opinion, GSEP is the gold standard for an intestinal metaplasia cohort. In this study, they recruited 3,000 high-risk Chinese Singaporeans, 50 years of age or greater, and followed them for at least 5 years with endoscopies. In this first study, published by Huang et al. In Cancer Cell in 2018, 203 samples from 148 individuals, 105 being high-risk intestinal metaplasia, these were OLGM 2 or above, 22 mild cases of metaplasia, and 37 normal controls. They also had germline data from these patients, and they performed bulk DNA sequencing, methylation profiling, and SNP analysis. So some of the key findings from this study were that the overall mutation rate increases with intestinal metaplasia severity. However, even in severe IM, the mutational burden at about 2% was lower than for gastric cancer at around 6.5%. There is an increase in somatic copy number variants or chromosomal instability in severe IM. Most commonly, they found amplification of 8q, which contains the MYC oncogene, as well as strong c-MYC expression. There was relative shortening of telomere length in the intestinal metaplasia cases relative to controls. There was also certain recurrent mutations found, most notably in the tumor suppressor FBXW7. However, they did not find P53 mutation, which is interesting because P53 mutation is found in about 50% of gastric cancers. There was also a generally higher level of methylation seen in IM. They had a small number of patients where they could assess progression, in this case 62, and of these 62, 82, and 6 progressed. The rest either stayed stable or actually regressed. And compared to progressors had a higher mutational burden, shorter telomere length, increased aberrant methylation, and increased chromosomal instability. So this is one of the first data that suggests that genomic technologies could be used to perhaps enhance our clinical risk stratification. Over the last 5 years, there's been rapid advances in genomic technology. Previously, inaccessible or cost-prohibitive techniques such as spatial transcriptomics, single-cell sequencing, or single-molecule FISH can now be applied to epidemiologic studies. So we use some of these techniques to better understand the gene expression profile of high-risk intestinal metaplasia. This is a manuscript that I and my co-first author, Ignacio Wickman of Stanford, and Puck of Santiago, Chile, published in NPJ Precision Oncology earlier this year. And so what our goal in this study was to identify a panel of genes which could successfully discriminate between high-risk intestinal metaplasia and lower-risk intestinal metaplasia. So in this study, we defined high risk as OLGMs 3 and 4, low risk as controls or OLGMs 1 and 2. And so we used bulk RNA sequencing, spatial transcriptomics, single-cell RNA sequencing, and single-molecule FISH to narrow down this gene list. Here are some more details from this study. What we first did is we had a cohort of patients from Stanford, from the Bay Area in California, and we had both a discovery cohort and a validation cohort. In the discovery cohort, we found that, and this is bulk RNA sequencing data, we found 105 genes were selectively overexpressed in the high-risk OLGM compared to low-risk OLGM and normal controls. We had a held-out unseen validation cohort, and you can see that 100 of these genes, the genes seen on the right, in the right of the column, were successfully validated in the validation cohort. So this 100 gene panel served as the basis of our further experiments. What we wanted then to do was to identify genes that were also expressed in gastric cancer and which localized the metaplastic glands. So what we did is we had a cohort of H&E slides which were hand annotated by an expert GI pathologist, Jeannie Shen from Stanford, and so she meticulously drew out the glands. So you can see at the top there, this is an H&E slide where the metaplasia is marked, as well as normal components of the gastric gland, the base and the pit cells. And what we did is in each cell represents About each well represents about 50 to 100 cells, and you can run a PCR reaction in each of these wells. So we then overlaid each of these gene signatures onto the wells, and what we found is that a composite of 26 of these genes did a fairly good job of recapitulating the areas of metaplasia in the H&E. So what we were next interested in is understanding what cell types within metaplastic glands were expressing these genes. There are many different cell types in metaplasia. There are goblet cells, there are enterocytes, there are immune cells. So which were the cell types that were expressing this gene expression profile? So we used single-cell RNA sequencing to answer this question. And what we found is that the gene signature was expressed by two distinct cell types. Now, the figure is a little bit small here, but you can see that here on the left, these are differentiated intestinal cells. So these contain, for example, enterocytes. These are abnormal cells. You shouldn't see enterocytes in the stomach. But we also saw a different population of cells which had more stem-like features. They were more dedifferentiated compared to these mature intestinal cells, yet they also displayed features consistent with intestinal or duodenal phenotypes. So we labeled these cells collectively intestinal-like stem cells. In the final step, what we did is we used single-molecule FISH. This gives us the ability to perform multiplex imaging on individual genes. And so we looked at both the markers of the differentiated cells, for example, goblet cells and enterocytes, as well as the intestinal-like stem cells. And what we found is that they were expressed in different compartments in space, different compartments within a metaplastic gland. So the goblet cells and the enterocytes were located in one area, and a separate group of cells which were smaller and had prominent nuclei were located in a different location within the metaplastic glands and expressed these markers of dedifferentiation. So we think that this is what these represent intestinal-like stem cells. Now, it's not just us that had this finding. The second study from the GCEP cohort was published in Cancer Cell in 2023, and they also found a dual population of cells in intestinal metaplasia. You can see from their single-cell sequencing here that there were both intestinal stem cells as well as these intestinal enterocytes and goblet cells. And here in the UMAP projections, you can see that here are the intestinal stem cells, and here are the enterocytes. And so what they did here is trajectory analysis. So they looked at how would the gene expression profile have to change for one cell type to go and become another. And so the distance between these intestinal stem cells to the enterocytes was much greater than the distance between intestinal stem cells and early gastric cancer. So this raises the very intriguing possibility that some have brought up before, that what we see as metaplasia, goblet cells and enterocytes, could actually just be innocent bystanders. It's really these intestinal stem cells that are the culprit and the cause of the gastric adenocarcinoma. So this also raises the possibility of using stem cell biomarkers for cancer risk progression. There was actually a little bit of data in the second GCEP paper that showed this. So in their second paper, they had more subjects, 700, and they followed them for 5 years. And in this case, they had 50 cases of dysplasia. And so what they wanted to compare was clinical risk stratification with clinical plus genomic risk stratification. So what they found is that Olgem stage, advanced age, pepsinogen index, and smoking were clinical features associated with progression onto dysplasia. And the AUC for this model, the clinical only model, was 0.71. But with the addition of certain pieces of genomic data, such as mutation count, clone size, and markers of chromosomal instability, they could increase this prediction to an AUC of 0.85. So this suggests, again, the possibility that use of genomic data can enhance our current clinical risk stratification systems. I wanted to shift focus here to talk a little bit about Helicobacter pylori. Now, eradication of Helicobacter pylori is universally recommended by US and international guidelines in patients with IM. Therefore, all intestinal metaplasia that comes to clinical attention is functionally Helicobacter pylori negative. Yet, even after HP eradication, patients remain at elevated risk compared to the general population. Moreover, there is some observational data and modeling data to suggest that eradication of HP may not substantially modify subsequent risk in GIM. So why is this? How can Helicobacter pylori negative cancers develop? And are there molecular biological features that can be used to risk stratify HP negative patients for cancer development? So the first thing I wanted to state is that perhaps negativity, clinical HP negativity, is not what it appears. We know that after HP eradication, recrudescence with genetically identical organisms may occur in 15% of individuals within 5 years. So that means clearly that they were not successfully eradicated the first time. Moreover, numerous groups have reported that patients who have been treated with antibiotics and have tested negative by conventional detection such as biopsies, urease tests, breath tests, or stool antigen tests will demonstrate persistent genomic material. This figure on the right is from my colleague and collaborator Manuel Amieva at Stanford. And so what he did here, his group did here, was he labeled Helicobacter with GFP, and he infected mice with GFP, and then he treated them with amoxicillin. So you can see, compared to the pre-amoxicillin figure, the post-amoxicillin figure demonstrated a 3-log reduction in HP burden. But a 3-log reduction is not eradication. As you can clearly see, there are residual HP organisms left. Perhaps even more concerningly, the antibiotic treatment changed the location of Helicobacter pylori. They went deeper into the glands, almost as if they were hiding. And deep in these pits, these bacteria were interacting with LGR5 positive stem cells located in the gastric glands. So this is from mice. What about in humans? Is there anything to this concept of molecular testing for HP eradication in humans? And I think there is some data to suggest that this is a real phenomenon. The figure on the left is a very nice study published from a group in Spain about 5 years ago. And so what they did in this study is that they compared conventional testing, that is histology with Giemsa stain, rapid urease test, and a urea breath test, with a highly sensitive molecular marker of detection, droplet digital PCR, on dyspeptic patients. And so they stratified patients by the number of positive conventional tests, 0, 1, 2, and 3. So the bottom column, which I've marked with brackets, are patients who tested negative, dyspeptic patients who tested negative for HP by histology, urease testing, and breath testing. So these were triply negative individuals. And then what they did is they targeted 3 genes. They had targeted VAC-A, urease, and the 16S subunit of Helicobacter. And they found that 36% of these patients had PCR products for Helicobacter pylori. So really, a lot of the patients that we think are HP negative may not actually be HP negative. Now, what about the application to intestinal metaplasia? There is some data from this in the first GSEP paper. So they had 120 HP negative histology by histology subjects, and of these, 28% or 33 subjects had Helicobacter reads. Moreover, they found that the higher the read density, the increased amount of chromosomal instability. Moreover, they found that extensive IM, that is involving both the antrum and the body, had a higher read count compared to antro-only disease. There's one other potential reason why cancer and inflammation can persist in HP negative stomachs, and that is that there are other bacteria that can cause inflammation. Specifically, I wanted to focus on the role of oral species. There is a known epidemiologic association between poor dental health, for example, caries, periodontal disease, tooth loss, and the risk of esophagus and gastric cancers. Moreover, certain pro-inflammatory bacterial species, for example, Streptococcus aeruginosus, which normally reside in the oral cavity, have been demonstrated in mice to induce gastritis, atrophy, metaplasia, and dysplasia, and have been detected in esophageal cancer tissue. There also appears to be a reduction in microbial diversity with advanced gastric inflammatory stages, which is independent of Helicobacter pylori. So in terms of human data, the study on the left was a 16S-based study from 81 cases, including superficial gastritis, atrophic gastritis, intestinal metaplasia, and gastric cancer from Xi'an, China. And what this group found was that 5 oral taxa demonstrated that combination of 5 oral taxa could significantly distinguish between various inflammatory states in the stomach. And when they took these 5 oral taxa and they applied it to a validation cohort, held on validation cohort, they found that it demonstrated good discrimination in AUC of 0.82 for gastric cancer versus superficial gastritis. We performed a smaller study, which was presented at DDW a couple of years ago, where we looked at 96 Helicobacter negative individuals from the San Francisco Bay Area, and our cohort was enriched in cancer dysplasia and intestinal metaplasia. And what we found is that relative to controls, cancer, dysplasia, and intestinal metaplasia, all of the differentially abundant species, or many of the differentially abundant species, were inflammatory oral bacteria. So I think that this idea that oral bacteria may play a role in inflammation and carcinogenesis in Helicobacter negative subjects is intriguing both from a diagnostic and a therapeutic perspective. From a diagnostic perspective, that means that perhaps some information derived from saliva could potentially inform the inflammatory status of the stomach. And from a therapeutic perspective, it suggests that perhaps improvement in oral health could somehow alter the gastric microbiome. So this is my last slide, gaps in biological knowledge. I think one gap is the integration of molecular data into clinically actionable tests. The NGS data that presented before is really currently impractical. It's cost prohibitive for most hospitals in the United States. So can we somehow use that data to develop a more clinically actionable test, such as a protein-based marker? The integration of genomic data with TCGA data, for example, does IM mutational phenotype predict gastric cancer subgrouping? The developing better non-invasive screening markers for GIM. Probably 95% of patients with GIM in the United States don't know that they have GIM. And finally, an increased biological understanding of the immune microenvironment of autoimmune gastritis, the other major source of inflammation. I'd like to thank funders, National Cancer Institute, and the Division of Gastroenterology and Hepatology at Stanford University. Thank you very much.
Robert, thank you very much for that very illuminating talk. Like I mentioned before, we're going to hold questions till the end of the session. And so now it's my pleasure to invite the next topic, which is going to be, unfortunately, Rebecca Fitzgerald from Cambridge, UK, was unable to make it, but her colleague Masi Di Pietro has graciously agreed to step in. So, Robert,
thank you very much. It's
Massimo,
a pleasure to be here.
Thank you very much.
And apologies from Rebecca, who has an injury from a recent holiday. It's not always safe to go on holiday before a big conference. She's really sad not to be here, but yeah, bear with me for the next 20 minutes. Actually, let me put my timing, so I'm sure that I'm not going to run over. So these are the conflicts from Rebecca. I don't have any conflicts myself related to this talk. It is commonly accepted that Barrett's esophagus is the only pre-malignant stage to esophageal adenocarcinoma, and it often transitions to cancer via intermediate dysplastic stages. However, there are still many questions. We know that only about 40% of people with esophageal adenocarcinoma will report history of Barrett's esophagus. And also, it is known that gastroesophageal junction adenocarcinoma can occur without apparent Barrett's esophagus. So while the picture might seem quite straightforward, because we know that gastroesophageal reflux disease is common in the population, about 10% of people will suffer from GERD, and about 10% of people with GERD will present with Barrett's esophagus. and a small minority of them will develop esophageal adenocarcinoma. In fact, it's probably more complex than that. As we heard from the previous talk, regression sometimes is described. People flagged with Barrett's esophagus and might actually be then told not to have Barrett's esophagus. We said that cancer can occur without intermediate premalignant stage. And actually, the transition from Barrett's to cancer sometimes is very variable. Sometimes it takes many, many years of surveillance for them to develop an early cancer. Sometimes catastrophic events can occur, and advanced cancer present all of a sudden in people with Barrett's esophagus. So there are still a lot of questions. So how to identify the factors that can influence transition to cancer? So clearly there are genetic susceptibility elements, and we know that there are some SNPs associated to development of Barrett's as well as esophageal cancer, for example, genes involved in the development of diaphragm. There are clearly environmental factors, smoking, obesity, that can influence the development of Barrett's and cancer. But more and more understanding is being gathered about genetic and genomic events, as well as interplay between epithelial and non-epithelial cells that can influence the transition from Barrett's to cancer. So this is a study that I was actually involved in as a gastroenterologist, although not in the fine molecular experiments. But one of the hypotheses that was brought some years ago is that Barrett's esophagus might originate from submucosal glands. And our first group was the first to actually isolate submucosal glands from esophagus. And via single-cell RNA sequencing, we actually found that submucosal glands here and non-epithelial here were actually very far from Barrett's esophagus. Most of the Barrett's esophagus cluster actually with normal gastric epithelium. These are biopsies taken from the stomach and the gastric cardia. So we did intensive molecular profiling using methylation, chromatin accessibility, as well as whole genome sequencing. And what actually came out of this is the confirmation that normal gastric in light green and Barrett's in darker green always clustered together and away from normal esophagus and submucosal glands, really highlighting the possibility that Barrett's esophagus does originate from gastric cells. So how does that work? We heard from the previous talk the involvement of MYC. Using organoids, we were able to actually identify that c-MYC was one of the oncogene that could have been involved in the transition from undifferentiated gastric cells into Barrett's undifferentiated and divided cells. And HNF4 alpha was a transcriptional factor that linked to MYC could actually reprogram the cells to Barrett's cells. And this is not unlike what happens in the stomach. In the stomach also, there was evidence that HNF4 alpha can bind cis elements in the CDX2 promoter near the CDX2 gene to actually activate the transcription of CDX2, which in turn can reprogram the cells to activate intestinal factors. We took a step forward and looked at the non-epithelial component, and here really we assessed carefully biopsies taken at different levels across the gastroesophageal junction. So gastric body biopsies, gastric cardia biopsies, biopsies at the squamocolumnar junction, as well as Barrett's, and normal. And what we found here is that the type of fibroblast that are expressed at different stages is different. We were able to identify 3 different types of fibroblast signatures that are called S1, S2, and S3. And what we found is that gastric metaplastic esophageal biopsies, which is what we think is the stage prior to intestinal metaplasia, had enrichment of S1. Whereas Barrett's intestinal metaplasia and gastric intestinal metaplasia had enrichment of S2 and S3 fibroblast signature. So it's really appearing that it's not just the epithelial component which is driving the progression to Barrett's esophagus and potentially to cancer, but also the interplay between the epithelial cells and the stromal cells. So does it matter whether Barrett's esophagus has intestinal metaplasia? Well, it appears it does, because if you look at the progression of Barrett's with gastric intestinal metaplasia only, as opposed to Barrett's with focal intestinal metaplasia and a fully intestinalized Barrett's esophagus, the rate of progression is different. And this is progression to low-grade dysplasia or higher, or progression to high-grade dysplasia or higher. We were able to demonstrate that really the presence of intestinal metaplasia is what matters, whether this is a bystanding phenomenon, as Robert was mentioning before, or whether this is the actual origin of the cancer cells is still something to debate. From a genomic point of view, it was interesting that when we profiled a number of different tissues, including Barrett's esophagus, primary tumors as well as metastases, and we looked at the different gene signatures related to the different conditions, we found that Siggener17, which is one of the commonest genomic signatures related to esophageal adenocarcinoma, was actually present as early as in the Barrett's non-dysplastic stage. And these Barrett's esophagus were not just Barrett's adjacent to cancer or progressed, were also Barrett's that never progressed. So it looks like signature 17 is something that occurred as early as non-dysplastic and even never progressed in Barrett's. And the same was true for signature 13. There were other signatures like Apobeck and the aging signatures that were exclusively present in the adenocarcinoma stage and probably a hallmark of the genomic events that occur in the progression to cancer. This was one of the earliest studies published more than 10 years ago, but was one of the first studies to highlight the fact that P53 is really, as a single gene, the main player in the progression from Barrett's to cancer. We are looking here at never dysplastic Barrett's in gray, Barrett's with high-grade dysplasia in blue, and maroon is the esophageal adenocarcinoma. And as in the GIM mentioned before, also in Barrett's esophagus, mutation of P53 is a very uncommon event. And it does happen in the pre-cancer stage, dysplastic pre-cancer stage, as often as in the cancer. So Barrett's with high-grade dysplasia have a high probability of having P53 mutation. There were a number of other cancer-related genes that in fact could not fully distinguish between pre-invasive dysplastic Barrett's and adenocarcinoma. And really the only one that seemed to mark the step from dysplasia to cancer was MAD4. And many others, like for example ARID1A, could also be found in non-dysplastic Barrett's. So this paper is under review, is unpublished, and this is the most recent combination of epidemiological and genomic factors which have been studied in relation to Barrett's and esophageal adenocarcinoma. You might be familiar with one of our previous early study where we found that esophageal adenocarcinoma with adjacent Barrett's has better prognosis compared to esophageal adenocarcinoma with no endoscopic evidence of Barrett's esophagus. So in this study, we really looked at epidemiological factors like age, smoking, BMI, heartburn, as well as a number of other genomic and gene level events. What we found here, comparing Barrett's positive esophageal adenocarcinoma in the middle and Barrett's negative esophageal adenocarcinoma in the left, is that many risk factors like smoking and BMI were not really different. There was a slight preponderance of heartburn in patients with just Barrett's esophagus and Barrett's related esophageal adenocarcinoma. When we looked at individual genes, what we found, and this is something that is coming recurrently in the recent literature, is that CDKN2A was commonly mutated in non-dysplastic Barrett's and mutated to a lesser extent in cancer. Whereas again, P53 was much more recurrently mutated in cancer compared to pre-invasive Barrett's esophagus. Other mutations were common between the Barrett's positive and the Barrett's negative adenocarcinoma, as well as other genomic events like whole genome doubling and tumor mutation burden. The signature 17 again was found as early as in the Barrett's. I'm going to skip this in the interest of time, and I'm going to go on to the issue of P16 CDKN2A gene, which is a really interesting story. So there's really discordance among studies on the rate of P16 mutation and whether P16 is involved in the risk of cancer in Barrett's esophagus. And you can see here in this study by Stackler and collaborators, the rate of CDKN2A mutation was similar in progressive and non-progressive Barrett's. The same was true in this study. And this is really one of the very few studies where, looking at CDKN2A, the progressors had a higher rate of mutation in this gene compared to non-progressors. Now, this is a recent study from Francesca Ciccarelli at QMUL, and that seems to settle the debate. Here we are looking at esophageal adenocarcinoma. You can see the high rate of mutation of P53, but CDKN2A was mutated at a much lower rate compared to P53. Here we are looking at Barrett's that progressed to cancer. And the rate of mutation of CDKN2A, even though slightly higher, 44% compared to 25%, was still not a very common event. And when they looked at pair Barrett's and cancer, actually the clonal relationship between CDKN2A mutation in Barrett's and subsequent cancer was very little. They found relation only in 23%. So it does not look like CDKN2A mutation is something that leads to cancer or increases the risk of cancer in Barrett's esophagus. And it looks like it's rather a protective factors. If you look at actually the rate of CDKN2A mutation in non-progressive Barrett's, actually is much higher, 62% compared to 44% in the progressed Barrett's and 25% in cancer. They did also find some cancer-related potential role of CDKN2A. So if that mutation happens later on, it might influence the cancer prognosis, and in fact, it seems to be related to worse prognosis of the cancer. But as far as the Barrett's is concerned, it rather seems to be more like a protective factor. There's an awful lot of copy number alteration in cancer compared to the adjacent Barrett's, suggesting that really this number of events occurs in the progression and does not fully precedes the progression. And really, we focus here on non-progressive Barrett's versus Barrett's progressing to esophageal adenocarcinoma. What we found again here, loss of 9P, the locus where CDKN2A was very common in non-progressing Barrett's. But at the gene level, in Barrett's that progress to cancer, really, P53 is the key player, together with loss of 17P, which is the locus where P53. And it seems then to drive all the other genomic events that occur in Barrett's, including, for example, whole genome doubling. Whereas in this study, we did find mutual exclusivity between P53 and ARID1A mutation. Another thing that we particularly were interested in is oncogene amplification in extrachromosomal DNA. And here we looked at two different cohorts from Cambridge and Seattle. What we found in this gene is that extrachromosomal DNA can be found as early as in non-dysplastic Barrett's, and it's very common in Barrett's at the stage of dysplasia. And in this particular timeline of one particular patient, you can see a patient that was found to have high-grade dysplasia, but at this stage, no extrachromosomal DNA was found. And then during the progression, this is a historical case from Seattle where radiofrequency ablation was not used. Then at subsequent endoscopy, there was one type of extrachromosomal DNA that we call 1. And a second type of extrachromosomal DNA happened when the patient progressed to cancer and was eventually treated. And this extrachromosomal DNA contained two particular genes, SOX1 and C2TA, which are involved in immunosuppression and evasion from cytotoxic T response, suggesting that extrachromosomal DNA could be another molecular event leading to predisposition to cancer. I'm going to skip on this, but just to say that methylation does not look like something that appears to provide a signature of high risk to Barrett's esophagus. And more as an overview, therefore, we can say that the early events that happens at non-dysplastic stage, like for example loss heterozygosity at 9p, might not necessarily provide the risk, but the main players is really p53 loss, which seems to be then driving aneuploidy and other chromosomal events. And really at the stage of transition to cancer, acquisition of extrachromosomal DNA and SMAD4 seems to be the step changes. And finally, I want to just mention that how can we make this actually clinically applicable? How can we design a test where we can, in an expensive way, predict the risk? And really, in these studies, we focus on shallow whole genome amplification. And in this longitudinal study, we took a cohort of patients that progressed and a cohort of patients that did not progress. And already at the non-dysplastic stages, we could see that the level of copy number alteration that you can see in patients that progress to cancer was higher compared to patients that did not progress. And you can see that there is a lot more red here because these are all the copy number events that happens at non-dysplastic stage in patients that will develop high-grade dysplasia or cancer later on. And this can happen as early as 5 to 8 years prior to progression. This is the end point, and here is looking backward in time. And you can design a test whereby with a decent area under the curve, you can predict progression to later stage. Eventually, how can we make the assessment of the Barrett's comprehensive? Well, one of the problems, actually, I'm going to refer to this study when we looked at the clonal heterogeneity, and we found that actually it's very heterogeneous Barrett's in terms of clones. So it's really difficult to represent, even with extensive Seattle protocol biopsies, the alterations in the DNA. But if we design a panesophageal sampling, and this is the example of a capsule sponge that can collect 360 degrees esophageal cells, then you can really identify early genomic events that can occur in preneoplasia and stratify the patients. I'm just going to mention this study where we use a combination of clinical factors like length of Barrett's and sex, together with simple tests like assessment of atypia on epithelial cells at p53. And we were able to re-stratify the patients with a really high accuracy, whereby patients with low risk had a very low chance on capsule sponge samples, low risk on capsule sponge samples plus clinical factors had a very low risk of bearing high-grade dysplasia cancer, whereas the prevalence of advanced neoplasia was much higher in patients with high risk. And just to mention that this study will be presented in plenary on Sunday by Keith Dunn, one of our PhD students, where we looked at the ability of capsule sponge to then go into the surveillance of these patients and risk stratify patients based on their risk to have dysplasia, to inform whether they need an endoscopy. And again, you have the classification in low risk, moderate risk, and high risk, tier 2 and tier 1, based on whether they have both tier 1 P53 and atypia, or tier 2 either P53 or atypia, with moderate risk just based on clinical factors, and low risk with all negative. And you can see clearly that the separation of these patients is impressive. Patients at high risk have a much higher chance to have dysplasia, and therefore should be followed up endoscopically, as opposed to patients with low risk that perhaps could be followed up just with cytosponge only. So just to conclude, Barrett's does occur to reprogramming and acquisition of an intestinal phenotype, although how this reprogram is triggered by reflux is still something that we are not able to fully understand. Loss of P53 is the commonest early event in the transition from Barrett's to cancer, whereas loss of P16 might even be protective. And finally, perhaps use of clinically friendly assays like shallow whole genome sequencing, perhaps in combination with non-invasive sampling, could be the way forward in terms of monitor and risk stratify patients with Barrett's. Thank you very much for your attention.
Thank you, Massimiliano. Please stay, come up and join us. And thank you again for filling in for Dr. Fitzgerald. So next I'd like to introduce Dr. Bob Coffey from Vanderbilt University.
Okay, I'd like to thank the organizers for the opportunity to present today. I'm going to take the prerogative of telling you about one protein that I'm particularly interested in terms of the progression of colorectal neoplasia, and that is dipeptidase 1. Having a little trouble here advancing the slides. Let me see if...
Just a mouse.
Here we go. So there are several reasons why we're particularly interested in DPEP1. And one is because 11 years after Bert Vogelstein and Eric Fearon formulated the Vogelgram, in which is an epithelial-centric sense that there are an accumulation of genetic events that result in various histologic changes from normal to adenoma to cancer. And so they were then trying to decide what criteria would they use to study genes further that they had identified by SAGE analysis. So the criteria they set were that the gene had to be present in adenomas and cancer, theoretically not in normal, 20-fold greater enrichment versus normal, and either a secreted or cell surface protein, with the thought that they were interested in biomarkers and potentially therapeutic targets. And when they did that analysis, they found 6 genes that encoded proteins that met the criteria, and one of the 6 was dipeptidase 1. Furthermore, Ken Lau and our lab teamed up and found an immune exclusion signature in microsatellite stable colon cancer. So these were 4 genes that were preferentially upregulated in microsatellite stable colon cancer, and they were inversely correlated with the presence of cytotoxic CD8 T cells. And of these 4 genes, one was DPEP1, which is the most abundant protein in small extracellular vesicles or exosomes of a number of colorectal cancer cell lines. Now, DPEP1 is a GPI-linked glycoprotein, and like virtually all GPI-linked glycoprotein, it resides at the apical surface of polarized epithelial cells. I'm showing you in red DPEP1 at the apical surface of polarizing MDCK cells expressing a DPEP1 cDNA. Now, DPEP1 has enzymatic functions, and these include being an extracellular dipeptidase for processing glutathione and leukotriene. So it acts as a homodimer, and it will then, GGT will convert glutathione to glutamate and cysteine glycine. DPEP1 will then cleave that dipeptidase to cysteine and glycine, and also in leukotriene LTD4, there's also this cysteine glycine that can be cleaved, that results in LTD4 becoming LTE4 and glycine. Now there is, and this enzymatic function can be blocked by cilastatin. Several years ago, a non-enzymatic function of DPEP1 was discovered, and that was the systemic injection of LPS in mice resulted in the induction of DPEP1 in liver and lung endothelial cells, where it was shown to act as a receptor for neutrophils. And it also acts as a chemoattractant for neutrophils and monocytes. And there is actually now an L-salt peptide. These are the first 5 amino acids of a 16 amino acid peptide that binds to DPEP1 and attenuates the ability of neutrophils to bind to DPEP1. And this is actually being used clinically in COVID patients that are at high risk for ARDS or kidney disease. So we actually found, and I'm going to be describing work that was recently published in JCI Insight, that DPEP1 can be detected at the base of the colonic crypts. You can see the staining that's apical, and Sarah Glass in the lab was able to show that in collaboration with Nick Markin's lab and Emily Green, that if you activate canonical Wnt signaling like AXIN2, NAKET1, DPEP1 is also upregulated, although to a modest degree, but it may be a Wnt target gene. We see DPEP1 in 28% of adenomas, and this increases to 81% of colorectal cancers. And I think you can appreciate the apical luminal staining. Now, DPEP1 is associated with neutrophil elastase staining as a marker of neutrophils in colorectal adenomas. I think you can see, see if I can, if you can see the apical staining for DPEP1, and then in the right panel, you can see occasionally, especially in high power, neutrophils that are intercalating among the epithelial and moving into the lumen. So there is an association between neutrophil elastase and DPEP1. And we actually see a high concordance between DPEP1 and neutrophil elastase staining in colorectal cancer, which is seen best on the bottom slide where you have the apical staining for DPEP1 and neutrophil elastase in the lumen, akin to dirty necrosis, which we think being associated more with microsatellite stable colon cancer. So then we wanted to further understand this relationship between DPEP1 and neutrophils. And so we looked at a battery of colon cancer cell lines, and we found that SW620 expressed high levels of DPEP1 compared to its primary counterpart, SW480. And so then Sarah isolated fresh neutrophils and then added them to these cultures. And what I hope you can see is on the bottom is a plentiful number of neutrophils binding to SW620, but not in the case of SW48, and we quantified this on the right. We also proceeded to look to see if we might see some attenuation of the neutrophil binding to SW620s if we incubated them with the L-salt peptide, and that's what I'm showing in the left panel. And when we quantify that, we see a dose-dependent decrease. These are micromolar amounts, but these are the levels that was needed in the cell paper to block the interaction of neutrophils with the endothelial cells. So it's not a great antagonist, but it does seem to be able to do that. The next question we had is, what would be the tumor phenotype in the absence of DPEP1? So we created colonic tumors in wild type and germline DPEP1 knockout mice using AOM, 2 AOM injections followed by 1 5-day course of DSS. And we wanted to know what the tumor phenotype was. Our prediction was that we were going to see an attenuation of the tumors because our bias is to think that neutrophils are going to be tumor promoting. And so we were in for a big surprise. So what we saw in the wild type was what was expected. So these lesions are occurring in the distal colon. In this case, we saw an adenoma, which was what we saw in all of the wild type mice. However, in the knockout, uniformly there was a sheet of tumors in the very distal colon. And we could see adenocarcinoma and squamous cell cancer in the very distal portion of the colon. And the adenocarcinomas were mucinous, and they were actually invasive. And likewise, the squamous cell cancers were invasive, as shown here. These were evaluated by Kay Washington, our GI pathologist. And so then we did the obligatory single-cell RNA-seq, and we see the different cell types shown here. But what I want you to focus on is the clear differences in the cancers between the knockout and the wild type. And we also see a cluster of squamous cells that we see in the knockout, but not in the normal. So how did this happen? We have features of what we think are features of microsatellite instability. So what we have done thus far is to show that there is a marked decrease in MSH6 and MUC2. When we stain for MSH2, we see a patchy loss of MSH2 in the tumors. Likewise, now we wanted to look at what was the status of the neutrophils. And what I'm showing here in the wild type, we see plentiful staining for myeloperoxidase, whereas in the DPEP1 knockout, there's a marked reduction in myeloperoxidase. That is a reduction in the number of neutrophils. Interestingly, for beta-catenin, we think these are microsatellite stable colon cancers, and they have the cytoplasmic enrichment of beta-catenin. But beta-catenin is actually sort of enhanced at the base of the epithelial cells, but it's still membranous. And finally, we wanted to look to see what is the status of the CD8 T cells. There's actually some evidence that neutrophils can actually lead to the exclusion of CD8 T cells in a MMP9 release of neutrophils that will then activate latent TGF-beta to exclude the T cells. And what we're seeing is the loss of the neutrophils, but a dramatic enrichment of the CD8 T cells, akin to what one might see in a microsatellite unstable tumor. And I should say that these tumors also express PD-L1, which is highlighted in the bottom. So that led us to then ask the question, could what we thought were originally microsatellite stable tumors in the absence of DPEP1 are taking on a microsatellite instability phenotype, and might these tumors now respond to immunotherapy? So this is Chelsea Sievers in the lab, and this is the schema in terms of both the wild type and the knockout. We're giving the 2 doses of AOM, 1 cycle of DSS. At day 65, we confirmed tumors were present, and then at that point, we're giving twice weekly pembrolizumab IP for 4 cycles of this regimen, and then at day 105, which took place on Thursday, we take the mice down, at least the initial cohort, to see what response we got. And so this is now looking, these are very preliminary results, but we're very excited about them in that both males and females, we have the control wild type treated, the knockout control, and the knockout treated in this small cohort that looks like there is a decrease in, I would say, in what we think are the colorectal cancers. We don't see a change in the distal lesions, which are squamous cell cancers. And then when we look only at the females, we see an increased response. Obviously, these are preliminary results, and we need longer cohorts. And so I want to conclude by saying that I think we've shown that DPEP1 is preferentially upregulated in microsatellite stable colon cancer in contrast to microsatellite unstable colon cancer. We've established a role for DPEP1 in neutrophil binding in colorectal cancer. And by germline knockout of DPEP1, MSS-like tumors display features of MSI-high tumors, and preliminary data suggest they respond to immune checkpoint blockade. So going forward, we need to test this in a larger cohort, optimize immune checkpoint blockade to exclude an enzymatic action of DPEP1. We're in the process of making a better L-salt peptide, and we need to put the DPEP null tumors into syngeneic C57 black 6 mice to see the properties of those tumors implicating more the host response. And then we're in the process of generating a DPEP1 flox allele. So I want to acknowledge many people in my lab and collaborators at Vanderbilt, and Donna Sanger, who provided the DPEP1 knockout. I want to call particular attention to Sarah Glass, who really drove this work, and she recently obtained her PhD at Vanderbilt. So thank you for your attention.
Thank you so much, Bob. I'd like to invite onto the stage now Anirban Mitra from MD Anderson Medical Center, Cancer Center in Houston, Texas, who will be talking about pancreatic intraepithelial neoplasia. Dr. Mitra.
Thank you.
Do I just do I click on my name? How do I get this?
I think you need to.
Oh, start.
Yeah.
Start.
And
Thank
then
you.
you have to hit this again, I think. Right here.
Here. Okay. Thank you, Bob. I want to thank Dr. Coffey and Dr. Huang for inviting me to this session. Any of you in the audience who's taken part in a CPC knows that the pathologist comes in the end and reveals a diagnosis. So only fitting that the GI pathologist gets to speak in the end. I don't have all the answers, but I was really struck during the last 3 talks, excellent talks, I should say, by the commonality of some of the themes that emerge in terms of precancers, and sort of the lack of progression in everyone who has these precancers. So I think if there can be a grand unifying theme someday, that will be really very important. So I'm at MD Anderson at least for the next 4 months before I move to New York to join NYU Langone. So some of you may be my future colleagues in the audience. This is my disclosure. This is from a license long time ago at Hopkins, but I will not be discussing it at this talk. So very quickly, in terms of precancer lesions of pancreatic cancer, these have been described a long time ago, over 100 years ago. This is actually from hand-drawn images of a Dutch pathologist, Dr. Holtz, and you can see anyone who's seen these precancerous lesions, they look really very similar to what we now see under the microscope. So this was something that was known, even though their significance obviously was not known in 1905. It took over 100 years, and then since then we've obviously now categorized these pancreatic intraepithelial lesions, and we have nomenclatures for them, classification schema. Here is a whole bunch of consensus papers that are noted here that describe these classification schemes. And this is from a review from Laura Wood, who's sitting right here, about the 2 major precancerous lesions, the microscopic pancreatic intraepithelial neoplasia, or PANINs. And it's PANIN because the prostate guys got there first and took PIN, so we don't have PIN, we have PANIN. And then IPMNs, or intraductal papillary mucinous neoplasms. This is a cystic precancers. I will only be talking about the PANINs this morning. My IPMN talk will be tomorrow morning at 8 for those who are early risers. So, a very long time ago, let me see how I proceed out here. So we developed with just very simple vintage technology that you would find in a museum these days, a multi-step progression model of PANINs. This is from 2003. It's immunohistochemistry, autoradiographic gels. It's exactly the stuff you find behind a glass these days. But this showed us that RAS mutations were very common in early precancers of the pancreas, and then much like what has been shown in the context of, say, esophagus, Barrett's esophagus, you have later events like SMAD4 loss or P53 loss that are concordant with high-grade precancers and predate the incipient sort of onset of invasion. And then Mike Goggins in 2012, in a paper that was published in Gastro, and this was without the obligatory single-cell sequencing paper data that Bob talked about. There was a time when you could publish in Gastro without single-cell data. It's harder now. Just using simple PCRs showed that RAS mutations are almost ubiquitous in these precancers of the pancreas. And not only that, even in the lowest grade precancers, almost all of them carried these KRAS mutations. Again, suggesting that this is a nearly universal phenomenon. It's almost sort of like the initiating genetic event. So in the last couple of years, I would say 2 proverbial nuclear bombs have burst in the precancer scene. And I want to talk about both of them because I think they really put into context some of the discussions we're going to be having. The first is this paper from Laura, and that was published last year. And she's actually going to be speaking at the 10 o'clock session, for those of you who can attend it, where she did, along with Ashley Keeman and Denny Wirtz at Hopkins, this wonderful study on 3D modeling of precancers in the pancreas. And I put normal in quotes here because these are actually normal adjacent to cancer patients. So they're not fully normal, but they're still histologically normal. And what she found really blows your mind. There is tremendous multifocality of these lesions, almost 1,000 lesions per organ based on modeling studies. And this is actually a 3D printout of the sort of the ductal system of the pancreas showing all these pannin lesions. And they all harbor different RAS mutations and are not necessarily clonally related to each other. Again, some common themes here in the GI precancer universe. So this was the first, I would say, really major finding, saying there's not one pannin that becomes cancer. The pancreata of at least cancer patients are ridden with these precancers. The other sort of dramatic finding that was published actually a year earlier from Michigan by Eileen Carpenter, who's a gastroenterologist. I don't know if she's in the audience, but and then Marina Pasca-Di-Magliano and colleagues here showed that pannins are truly everywhere. There's a very high prevalence of these lesions in the pancreas of cancer-free organ donors. So they collaborated with this organ donor network called Gift of Life in Michigan, and they found that almost 60% of the cases they looked at, including 20-year-olds, had these precancerous lesions in the pancreas. And we've now looked at this in a couple of independent cohorts, one from Nebraska and one from UT Southwestern. I have looked at those slides myself, and yes, about 70% of pancreata in cancer-free organ donors harbor these precancerous lesions. This is a 23-year-old with pannins in the pancreas right here. This is a 67-year-old, and this is a pretty juicy-looking pannin. I mean, this is getting to high-grade levels here. But it is shocking how common these precancers are. In this room right now, there is a sizable minority of us that likely have pannins in our pancreas. So the question, fortunately, is why isn't everyone developing pancreas cancer? If this is so common, why is, thank heavens, the rate of pancreas cancer, even though it's going up, is still a relatively uncommon disease. And so if we just kind of elaborate some of the theoretical barriers to cancer progression, I can divide them into precancer cell intrinsic, precancer cell extrinsic, which is in the precancer microenvironment, or PME, and this is very distinct from the TME. I think the tumor microenvironment, which is what we are all typically used to studying in cancer biology, is actually distinct from the precancer microenvironment, or PME. That has its own sort of nuanced findings. And then you have the systemic influences, the macro environment. So all three, I think, are working in conjunction to sort of stem the progression to frank neoplasia. So in terms of intrinsic, RAS induces tremendous adaptive changes in cells. Once you have a mutant RAS in a cell, it's not the same cell. And some of those are actually mechanisms that block the progression to frank neoplasia. The presence or absence of co-mutations. Again, we heard this fascinating talk in the context of Barrett's, and this is also true in pancreas cancer. And then non-genomic transcriptional alterations, including things like ecDNA amplifications, etc. Altered immune and stromal composition. This is a very important finding, and I'll show you some data, recent data on this in the context of pannins, how things change very early in preneoplasia. This is not, we talk about, again, the TME in the context of established cancer, but changes are happening in the PME really early on, and some of them are permissive, others are restraining. And the balance, the seesaw balance between them is, I think, what plays a huge role in terms of the progression. I put intrapancreatic microbiome only because I was talking about the PME, but really, as again was shown in the context of the oral and gut microbiome in a previous talk, this is something that impacts at a distance. This is not something that necessarily has to be locally present, although it is to some extent. And then systemic influences, diet is huge, glycemic status, environmental factors. We have Dr. Chen sitting here in the audience who's doing this, has done really remarkable studies in the context of colorectal cancer predisposition. And so these systemic influences are really, really important. So this was, again, a very simple study done, a long time ago now, 16 years ago, looking at what happens when you have mutant RAS. And basically, when you have mutant RAS, there is tremendous replication stress that is induced in these cells. And that, in turn, induces replication stress response. Checkpoints like ADM and CHEK2 get activated in these cells. And that's one of the reasons why these pannins kind of sit there. They have barely any proliferative response. When you do Ki67, these pannins have very low proliferation rates until you lose P53. Again, you heard P53 before in the context of Barrett's. This truly is sort of the gatekeeper, and once P53 function is lost, you really unleash the progression to invasive neoplasia. And that's exactly what we see. So there's a lot of breaks that come in the moment RAS is mutated, and you need to lose P53 for those breaks to be malfunctional again. I also talked in terms of the intrinsic factors, the impact of the actual allelic variant, as well as co-occurring mutations. And I'm just going to show you sort of one example from some of the work we have done in the lab, showing both of these sort of variables and how they impact the rate and frequency of progression to cancer. So we have a model in the lab where basically it's a Cre-driven Cas9 that allows you to regulate Cas9 expression in any organ or cell type of interest, and then we use an AAV-driven carrier to introduce mutant RAS so that there's no RAS allele in the background of these mice. We introduce the RAS through homology-dependent recombination, as well as guide RNAs against pivotal tumor suppressor genes like P53, ARID1, LKB1, etc. And what this shows is, so this is data with just G12D as the recombination template. So we are introducing at the endogenous locus of RAS in the mouse pancreas, a G12D. And this is not done like the traditional Cre-driven model where you have the entire pancreas expressing a mutant RAS and then you get a cancer. So this is very localized, and this kind of models what we see in humans. It's not that the entire pancreas has RAS mutation. There's a small focus, and that then develops into pannins and eventually cancer. So what we see here is these RAS mutations, when you just have them by themselves, you get pannins, but you really don't get cancer after following these mice for a long time. And then you start introducing co-mutations, and some of them just also give rise to pannins only, like this ARID1A co-mutation. And then once you have P53, now you start getting cancers. You have P53 plus ARID1, now you start getting the same cancers, the same rate. The ARID1 really doesn't add a whole lot. And with LKB1, the same thing. So again, the co-mutations really matter. RAS by itself doesn't do much, but then once you start having things like P53, you start progressing to cancer over time. What about the actual allelic variant? Does the actual specific mutation in the RAS, whether it's D, V, R, does that matter? And this is, again, inspiration from some of the work that Laura did, showing that there are these pannins, multifocal pannins all over, and they have all sorts of mutations. Some are D, some are V, some are R. In fact, the photograph I showed before from her paper has R mutations all over. So it's very hard to make a G12R model. Many folks have tried. R is a very weak allele. It doesn't form cancers typically, even though it's a third most common allelic mutation in human pancreas cancer, but in the mouse it doesn't. And so when we replace the D with the R, we observe no phenotype, not even pannins, for over a year of follow-up. And then we put in P53. Remember, these P53 null Ds all got cancer very quickly, 100% penetrance. But here, very low penetrance, took us 9 months to get there. And then we put in LKB1 on top of the P53, and now suddenly you start getting cancer. So what I'm trying to say is, just having a RAS mutant pannin may not be enough. Stochastically, you're going to need a second hit, a third hit potentially, depending on the mutation. So it takes time for these pannins to become full-fledged cancer, and probably one of the reasons why, thankfully, so many of us have pannins but don't get pancreas cancer. So in terms of the precancer microenvironment, or PNE, of pannin lesions, there's two really nice papers that have been published in the last couple of years. First is, of course, the paper from Eileen Marina and Tim Frankel at Michigan on the donor pancreas data, and then the second one from Hopkins from Luciani Kawahara and Ilana Fertig in Cell Systems, looking at sort of the spatial and single cell distribution of the microenvironment around these precancers. And what did Luciani and Ilana find? First and foremost, many of the stromal fibroblast subtypes like myofibroblastic calves, inflammatory calves, and antigen-presenting or AP calves are present. These are calves that have been described cancer-associated fibroblasts. By definition, the C stands for cancer-associated, but they do occur very early on, even in the state of precancers. In fact, in the earliest low-grade precancer. So maybe we should really rethink the acronym CAF, because they're not really always cancer-associated. And what's very interesting, again, always look at the supplementary data. The supplementary data has beautiful figures. In the supplementary data, they had this spatial data showing how CD4 cells align and are spatially localized with these antigen-presenting CAFs, suggesting, remember, as the name suggests, antigen-presenting CAFs present antigens in the context of class 2 HLA. And so there may be some education going on of these CD4 T cells here from the antigen-presenting CAFs, thereby likely sort of launching a productive immune response against these precancers. Again, remember, it's a seesaw battle, permissive versus restraining, and this AP-CAF CD4 interaction may be one of the restraining influences that sort of is caused by the host immune system and prevents these precancers from going forward to full-fledged cancer. Same thing was observed in this data from the Michigan group. Again, lots of CD4 cells showing up in these pannins, and the CD4 cells are not Tregs, so they're not regulatory T cells. They're again likely cells that are involved in sort of the class 2 priming and productive immune response against these early precancers. And the sum total of all of this is that as these pannins progress to cancer, not surprisingly, the inflammatory milieu that we see early on goes away and is replaced by a proliferative milieu, okay? So early on, there is a lot of immune response that the body has against these precancers, but over time, that's gone. So we asked ourselves this very simple question, which is, are molecular signatures of cancer-associated pannins distinct from organ donor incidental pannins? I mean, they're kind of really different situations. One is there's no cancer in the pancreas. These are individuals who unfortunately passed away from something else, like an accident or a gunshot wound. And then you have these other pannins that are arising in a pancreas that has cancer, and are they different? So to address that, we did a study where we took individuals, pancreatiform individuals who had pancreas cancer, and then a series of individuals who had pannins in the context of organ donation. And we did very specific AI-guided histology analysis and selection. So I was kind of the ground truth here as a pancreas pathologist. And we worked with a company called Omic Vision, which is a Danish company, and they have developed a very nice AI tool. And so we kind of worked together, looked at hundreds of slides, and developed a model that allows AI to basically pick up these lesions, pannins, acinoductal metaplasia, normal ducts, cancer. So yours truly was sort of the ground truth pathologist on this. And then we deployed something called deep visual proteomics to look at the protein signatures of these cancer-associated and incidental pannins. So what is deep visual proteomics? This is a technology that has really come to the fore in the last couple of years. It has 4 steps. One is you do high-resolution microscopy and image analysis, and this is amenable on FFP archival sections, so you don't need fresh sections for that, and therefore it opens up remarkable possibilities of using retrospective samples. You have foundation models that use AI-powered pathology, like we developed here, laser microdissection of precisely defined phenotypic cells. Only 100 cells are enough for us to get thousands of peptides using ultrasensitive mass spec methods like this EvoSep Astral that is used by Omic Vision. And this was first featured by this foundational paper in 2002, and they've really done some remarkable work over the last few years, not all in cancer, and it really were sort of also highlighted as a method of the year by Nature Methods last year. So this is what we did. So this is your AI-guided computational pathology. This is actually pancreas cancer. I'm showing this sort of as an example because we have another study in the context of cancer metastases ongoing with them. And this is what it looks like the AI model that they've developed precisely outlines areas for LCM capture and mass spec proteomics. You can see the cancer is nicely highlighted here, and this is exactly what we did with the panels. Again, I checked everything to make sure what they were doing was really true, and it is. So basically, we ended up analyzing about 93 individual lesions across 16 patients that were microdissected for mass spec analysis. So what did we find? So deep proteomic landscapes or DVP, deep proteomic landscapes of incidental and cancer-associated panel lesions. I basically asked them to classify these as incidental pannins or I pannins and cancer-associated pannins or C pannins. So you're going to see this term show up in the data. And again, that was sort of an easy way of differentiating this between them. The important thing is, again, from these 100 microdissected cells of these lesions, we were getting close to 5,500 proteins on average, which is really remarkable. I have not seen data like this. And overall, we identified about more than 8,500 individual proteins from this data set of 93 microdissected lesions. So what did we find? This is a simple sort of PCA analysis. I don't know what happened to the incidental pannins. That's the one in yellow. I'm sorry about that. Somehow it got missing. But the bottom line is, when you look at these incidental pannins, and I normals are the normal ducts associated with these incidental pannins in the organ donor samples, they kind of cluster separately from the cancer-associated normal ducts and pannins. Not a surprise. The normal ducts in these organ donors are also distinct from the pannins. You can see there's a whole spectrum of pannins that some are closer to these normal ducts, some a little bit far away, and then these orange blobs are the ADMs that we microdissected as well, and they are also kind of like a bridge between the normal and the pannins. And then the cancer-associated pannins and normals are quite distinct, okay? These are all the cancer-associated normal ducts, and then these are the cancer-associated pannins. A few of the organ donor pannins make their way into this, which is kind of interesting, and we'll have to figure out what that means for the individuals involved. So DVP allowed us to identify both core and distinct signatures of these I pannins and C pannin lesions. These are some core pannin signature proteins that were not distinct irrespective of where the pannin was coming from. Things like S100P, Claudin 18, MUC5AC, and Exin10. These have all been described by other groups as well as sort of a common pannin signature. But once you look at this globally in terms of the proteomic landscapes, it's very clear that cancer-associated normal and pannins are very distinct from incidental pannins and incidental normal ducts. I mean, again, it's hard to see the individual proteins here. All of this data will be in bioarchives very soon, so don't worry, you can take a look at it. But bottom line is, you can see globally at the protein level, these look very distinct. That's somewhat different from the data at the RNA level from Marina and others, Eileen Carpenter's paper that was published in 2023, where they did not find major differences at the RNA level between these organ donor pannins and cancer-associated pannins. But again, RNA and protein are always not in sync, so I'm not really surprised. The one thing that's very interesting and that's in common with prior work is that these pannins are like gangbuster of the classical lineage. And now in pancreas cancer, we have two major lineage subtypes: the classical lineage, which are better differentiated, mucinous lesions, are more responsive to chemotherapy, and then you have the more aggressive basal lineage of cells, which tend to have mixed signatures, etc., are typically refractory to chemotherapy. And you can see here, pannins, both the incidental and cancer-associated pannins, are massively of the classical lineage, in contrast to the cancer cells, which in turn tend to be more of the basal lineage subtype, here as marked by HMGA2, which is a very strong basal marker. So what are the signatures that distinguish these cancer-associated pannins from incidental pannins? Again, there's a lot of differences between them. They're not alike at all. And if you look at some of the top things that show up, there's interferon response signaling. That is probably one of the most important differences. There is and this is maybe not a surprise given some of the data on the immune activity that's going on here. T cells are basically able to come in and maybe stimulate this interferon response. There's a lot of ER stress that these pannins are in. massive metabolic alterations. The one we found that was striking, and I'll show you the next slide, showing that this is actually one of the earliest changes in cancer, is that glycolytic genes like PGAM1 go up, and then PTK4 goes down. And PTK4 is actually an inhibitor of OXPHOS, and maybe there's just a general regulation of metabolism, but PTK4 also has a role when it is inhibited in enhancing lipogenesis. So I think there are massive metabolic perturbations that are happening in very early cancer that would be worth studying functionally. And then upregulation of vesicular trafficking proteins. So this is looking at, and again, this blew my mind, these are histologically normal ducts, absolutely normal, nothing in them. And if you compare these incidental ducts versus cancer-associated ducts, major differences. Many of them, the ones that show up in the pannins, and of course all the way in cancer as well, suggesting that these things may be baked in, these changes may be baked in pretty early on, even in the context of normal ducts. Again, for example, PTK4 going up or going down, depending on which direction you look at in the cancer-associated normal ducts, PGAM1, which is a glycolytic gene, going up. So, very quickly, this is my last slide. Pannin lesions are widely prevalent in the general population, almost 60%, including in young adults. 20-year-olds have pannins. Factors restraining pannins from progressing are likely multifactorial. Pannin intrinsic, like the specific allele or co-mutations, in the exo-PME or exocrine cancer microenvironment, and then systemic influences we didn't really talk about, but we have a lot of work ongoing in the lab in terms of these systemic factors. So deep visual proteomics, which is the data I just presented, identifies profound differences in the proteomic landscape of C pannins versus I pannins, as well as the corresponding normal ducts. And many of these changes we observe in the pannins and then subsequently in the cancers are baked in early on. The question then is obviously chicken or egg. Do these changes happen because these things are next to the cancer or because this is what the cancer arose in? And again, you need to do functional studies for that. Really to understand. But does this present an opportunity for intercepting cancer? Obviously is an area of high interest to many of us. Now that we have some of these targets, if we perturb them, what happens in the models that we have in our labs? So I want to thank my lab, the folks who did much of the work. Omic Vision have been unbelievably great collaborators. Again, we'll put this data out on bioRxiv soon. It's the largest proteomic data set of any pre-cancer, so go to town. I want to thank UNMC collaborators who provided much of the incidental organ donor pannin data sets to us. And then NCI Consortia TBEL that Bob and Laura are both part of. We are doing really, really great science in early lesions through this consortium. And PSRC, which is another consortium that I'm part of in pancreatic cancer stroma. So thank you all very much.
Dr. Mitra, thank you so much. Please join us on stage. So we are out of time in the session, but I think the speakers will be available after if you wanted to come up with any additional questions. We'll stay a little bit longer here. Thank you all for your kind attention.